All right, everyone. Today we're going to be talking about the visual system. And so in this image that you see here, you're just looking at some tractography that was conducted. So what you're seeing are the fluoresced fibers that comprise the visual system. So I just put that here because it looked really cool, honestly. So what we're going to talk about today, we'll briefly mention the learning objectives, cover some introductory information about the visual system. Then we'll look at the retinogeniculostriate and extrastriate pathways in detail. So we'll talk about the parvocellular and magnocellular pathways that comprise them. We'll look at briefly some brainstem slices. You won't need to know the detailed anatomy of those, but I'm just going to show you where those structures are located in the brain when you're thinking about the brain in different orientations. Then we'll talk about lesions along those pathways and what kind of deficits to vision you might see with those lesions. And then at the end, we'll talk about the targets of non-lateral geniculate nucleus projecting retinal ganglion cells. So you do have some retinal ganglion cells. Those are the bipolar sensory neurons in your eye that are associated with vision. And so some of those cells actually have a different pathway that is not necessarily involved with visual perception, but it's actually involved with things like sleep-wake patterns, and then also we'll talk about the involvement of the pupillary light reflex. All right, so if any of you have ever been watching this show, it's on Disney Plus right now. I highly encourage it. WandaVision, it's really good. If you're okay with, paying a certain fee every month to Disney in order to obtain Disney Plus. So real quickly, for this lecture, you should be able to identify nuclei, tracts. Blood supply is not really all that important as much, but mainly nuclei, tracts of the retinogeniculostriate pathway and a variety of images. You want to compare and contrast the anatomical and functional contributions of the parvo and magnocellular pathways. Recognize visual field deficits as a result of lesions along those pathways, and then distinguish between those non-lateral geniculate nucleus projecting retinal ganglion cells that I mentioned earlier. All right, so let's start by talking about the retinogeniculostriate pathway. It's a very long word, but all we're saying is we're involving the retina and the retinal ganglion cells, the lateral geniculate nucleus in the thalamus, and the striate cortex, also known as the primary visual cortex. So it's just one long word, squashing together all those terms that let you know the main players in that pathway. So your retinal ganglion cells are those bipolar neurons in the retina, and they innervate the lateral geniculate nucleus in the thalamus. So how do those retinal cells get to the thalamus? Well, Their central processes actually synapse on cell bodies in the lateral geniculate nucleus, and those central processes are essentially the fibers that comprise the optic nerve, and then ultimately optic tract, and then from the optic tract, they then arrive on the lateral geniculate nucleus in the thalamus. Now, from the lateral geniculate nucleus, fibers will project. So these are axons extending from those cell bodies in the lateral geniculate nucleus, and then they will synapse on cells in the primary visual or striate cortex. Now, the name we give those bundles of axons that are going from the lateral geniculate nucleus to the primary visual cortex are the optic radiations. So the nuclei, which have the cell bodies, have names, and then the axon fibers that form bundles between these nuclei also have names. And in your primary visual cortex, this is where you have the beginning stages for visual perception. The visual field is an area of the environment from which light can activate our photoreceptors. So those are the rod and cone cells inside your retina. And so our visual field is perceived by both eyes. Therefore, you have a left and right visual hemifield for both eyes, because both eyes are seeing what's in front of you. All right, and so that will be important later. Now, if you look at this illustration, right is right and left is left. And so for those of you that have looked at radiographic images, I'm not sure if you've had to look at radiographic images before, but the rule of thumb is that everything is reversed. So right is left and left is right. But in this case, what you're looking at is an image as if I were looking directly at the top of someone's brain. So if I'm looking directly at the top of someone's brain, then left is left and right is right, per this visual diagram. Now, cross projections that are going to the primary visual cortex are going to occur in the optic chiasm. So the optic chiasm is that area where you see blue and red fibers crossing somewhat in the middle of the brain. We call that the optic chiasm. So this is what creates a hemifield representation in your primary visual cortex, because you have partial crossing of the visual field from both eyes. And that's ultimately going to go to the primary visual cortex. The retinal ganglion cell axons from the temporal parts of the retina stay on the same side. So if you look at your lateral aspects of your eyeballs, okay, so looking at this diagram, you see on the lateral aspects of the eyeballs, we call this the temporal area, okay, because it's around the near the temporal bone, you know, where the temporal lobes of the brain are located. So those fibers are going to stay on the same side. They will not cross. But the nasal part of your retina, so that part of your retina that's more medial, that's closer to the nose, those fibers will cross in the optic chiasm. Okay, and you can see that with the different color representations as well. All right, so this diagram is showing you how the visual field would look if we were really seeing this nice contrast separation of, what each eye is taking from the visual field. So as you can see here, you've got both right and left hemifields represented in both eyes. Now, that's not the end of the story. So not only do you have, let me back up for a second. Not only do you have partial crossing of information for right and left, so you have a partial crossing of left eye information going to right side of brain, partial crossing of right side information going to left brain. Okay. You also have a superior and an inferior distinction represented in your primary visual cortex as well. So information that is in your superior visual field will be going to the bottom of your primary visual cortex. Information in the inferior visual field is going to be going to the superior aspect of your primary visual cortex. And we're going to talk about why that happens in just a few minutes. All right, so you also have a distinction between macular versus peripheral distribution. So macular would be that high acuity vision as opposed to peripheral vision from the side. And they have different representations in the primary visual cortex as well. So you'll note that from my periphery of my eye, represented by the green and red, also in this image, they're going to the anterior or front part of my primary visual cortex. Now, if you follow the black line, which represents the macular or high acuity vision, it's going to the posterior aspect of your primary visual cortex. So, if you look at this diagram, right is left, left is right, up is down, and down is up. So one way to think about this is that because axons from right and left eyes partially cross in the optic chiasm like we saw earlier, and because our lens actually inverts the images that we're seeing, and we'll see that an example of that in just a few minutes. The right side of the visual field goes to the left side of the brain. The left side of the visual field goes to the right side of the brain. Also, because of the inversion of this image, because of the lens of our eye, top of visual field goes to the bottom of the primary visual cortex in the brain. Bottom of the visual field goes to the top of the primary visual cortex in the brain. All right, so now we're going to go into the more intricate details and specifics of these pathways. So let's think of the eye as a camera. And so because the convex lens of the eye inverts the image of what is being viewed, the brain has to turn that image back around and right side up again so that we perceive the images of objects and people as they really are. So that's why you've got this crisscrossy, inverted projection of fibers, because our brain has to turn the image back right side up so that we can see it as it really is. And a camera does something similar to this with the images that you take in a camera. All right, so as you can see, light is reflected by the lens and projected onto the retina, as we see by these crisscrossing red arrows. And the image is inverted at this point, so the retinal image is upside down. Okay, but as we said earlier, the brain's going to flip that back right side up again so that we can perceive what we're seeing right side up. And on your own time, you can watch this clip from the Wild Wild West where they flip the guy's head upside down. I don't know if you've ever seen that movie, but it's a nice clip. You can watch it on your own time. So we mentioned macular vision, so that high acuity vision. So that is situated in almost the central part of the retina in the back of the eye, where you also find the fovea. So both of these areas are for high acuity vision. Now, you've probably heard of a blind spot. You've heard of, you having a blind spot. Now, what is that? So that is the area where all of the central processes of your retinal ganglion cells are leaving the retina, leaving the back of the eye to give rise to the optic nerve. So all those processes, fibers, collect in that area, and there are no photoreceptor cells there. So because there are no photoreceptor cells there, you cannot detect anything in that area. And so that's why you have the blind spot. So let's take a closer look of the fovea. So the fovea is denoted by this depression. You can see it encased by this red rectangle. And the reason why it looks like a little pit is because all the retinal interneurons have been sort of moved out of the way, and they create this depression. And we only see this in animals like diurnal primates, that are active during the daytime. And then other organisms, very few birds and fish though, have also developed this due to convergent evolution. And so they also are active during the daytime. The macula is a thickened area, and it's lighter, hence the name macula lutea, because it gets relegated a lot more photoreceptor cells that allow those layers that have more cell bodies, that have more nuclei, to be thicker, and therefore you also have more fiber projections that are synapsing on those cells, and that also gives you a lighter appearance because you also have more fibers that are synapsing on these cells, thus giving it that thicker density. Now, the retina, as displayed here in both this illustration and this sample from a histological slide, you will see that light essentially has to pass through all of these layers. So if you look on the the right hand side of your screen at the histological slide, you'll see that light goes through all of these layers to then come into contact with the retinal ganglion cells. And those are our photoreceptor cells, okay? So our photoreceptor cells include cones and rods. The cones are for more high acuity and color vision. So guess what? We're going to see a lot more of those in the macula and fovea areas. The rods are more so for peripheral vision and motion. So guess what? We're going to see more of those, a higher concentration of those along the periphery of the retina. Visual perception starts to begin in the retina due to the processing of two different types of bipolar cells and their connections with horizontal cells via interneurons before they actually synapse on those retinal ganglion cells. So if you look here, you've got these bipolar cells that are in blue. They have the blue squares around them. And you'll notice that they have connections, synaptical connections with both the horizontal cells that are designated by the H and these other little interneurons that are designated by A. And so this cross communication will allow for information concerning visual perception to be relayed to those photoreceptor cells. Now, we were talking about high acuity and peripheral vision, and so that relates to this next topic where we'll talk about parvocellular and magnocellular pathways. So there are at least two types of retinal ganglion cells that exist for visual perception. You have the P type, which are the most abundant, and the M type. And these are going to participate differentially in visual perception. So you have this nice little table here that shows you their distinction based on where they are in the retina, the size of their receptive fields, their spatial frequency responses. So is it high? Is it a lot of detail and color? Is it low? Are we just looking for broad outlines? And then the temporal frequency response. So is the object standing still or is it moving? So both of these cell types and hence their pathways have these differential functions, and this table helps summarize that. So what might this look like? So if you take this image of this monkey, the magnocellular system, which is that one that's more of the peripheral vision, it's mainly going to be looking at broad outlines and movement. Okay, now if you look at the separated image that distinguishes that magnocellular perception from parvocellular, you'll see there's a lot more detail involved. And it makes sense that an object needs to be standing still for us to perceive that detail. So there, some retinal ganglion cells are photosynthesis sensitive. There are some that Dr. Saya may talk about later in his lecture on sleep and wakefulness, dealing with circadian rhythms. So we won't go into a whole lot of detail about that, but there's a slide toward the end of this lecture that kind of touches on it a little bit. Now, these different types of retinal ganglion cells, parvocellular, magnocellular, are going to terminate in an organized fashion within the lateral geniculate nucleus. Our magnocellular axons are going to terminate in layers 1 and 2. Okay, so we can see layers 1 and 2 depicted here in image C. So you're looking at a lateral geniculate nucleus in this image, image C. Our parvocellular axons are going to terminate in layers 3 through 6. So you have 6 total layers in that nucleus. So image D really shows you that representation of those 6 layers in an illustration. So we also have differences in retinal ganglion cells coming from the nasal portion of the retina and the temporal portion of the retina. So don't forget that. Remember that there's a crossing of fibers at the optic chiasm, and then there's a passage of fibers on the same side through the temporal region, those uncrossed fibers. So contralateral or opposite side eye representation is relegated to layers 1, 4, and 6. Ipsilateral or same side eye representation is relegated to layers 2, 3, and 5. And so that's your C's and I's are designating that contralateral or opposite side, and ipsilateral or same side designations in the lateral geniculate nucleus. Now, the lateral geniculate nucleus cells will then project back to V1, or primary visual cortex, also known as the striate cortex, in an ordered fashion. And so we've already kind of summarized the movement of these fibers, or the pathway of these fibers in the introduction. But here again, we have this image, and now we're going to look at those areas as they appear in these radiographic images that are showing you a sagittal section through the brain, as if you were cutting the brain into right and left sides. And so what I want to draw your attention to is the calcarine sulcus, which is this groove that separates the 2 portions of our primary visual cortex. Those are the cuneus. Cuneus comes from the word that means wedge, because it looks like a wedge. So that would be the upper or more superior portion of our primary visual cortex, so above the calcarine sulcus. And then the lower aspect of the primary visual cortex we call the lingual gyrus. So the lingual gyrus, because it kind of looks like a tongue. And then here again is your nice image that's showing your differentiation and fiber projections. So here's our lingual gyrus. The lingual gyrus is going to be receiving, so the lingual gyrus is actually going to be receiving the information from the superior visual field. All right, because the superior visual field information was, remember, inverted in the from the lens of the eye. And remember that central vision is relegated to the posterior aspect of our primary visual cortex in the lingual gyrus, as well as in the cuneus, as we see in this image. And so remember, our upper part of our primary visual cortex is receiving information from our inferior visual field, again, because that inversion of images from the lens of the eye. The Meyer loop is representing these more inferior fibers in the optic radiations. These were named after my great ancestor long ago. I'm just kidding. I don't know who the guy is, but his name is spelled like mine. All right, now it's all and well to look at radiographic images, but how useful is that to you? Because you're probably not, you may not be looking at radiographic images very often. So on an actual brain, let's look at those structures. So here we have the parieto-occipital sulcus. That's going to separate the parietal lobes from the occipital lobe. And we also have the calcarine sulcus, okay, that's dividing the two portions, upper and lower, superior and inferior aspects of my primary visual cortex. And here they are overlaying on these radiographic images below. The cuneus on top, and then the lingual gyrus on the bottom. Now, it gets more complicated. The primary visual cortex, or V1, can be broken down into columns. We have what are known as ocular dominance columns. So the lateral geniculate nucleus layers 1, 4, and 6, 2, 3, and 5 that we talked about earlier are actually going to project to a layer in the primary visual cortex called layer 4, that's Roman numeral 4, in specific columns. So you see these columns, white and blue, that are coming from these nicely differentially colored, same, in the same format from the lateral geniculate nucleus. It's just showing you that those layers are projecting to these ocular dominance columns in layer 4 in the primary visual cortex. On top of that, we also have orientation columns. So if you look at these overlaying columns, so look going down the sides in more of a longitudinal perspective on the left-hand side of this upper image, this upper diagram, you'll see these little lines that show the orientation in which the light is received by these cells, or the perception of the light and how it's received by these cells. And so all the cells within these columns are going to receive light, but in that orientation and at that angle. And so response properties of neurons within this retinogeniculo pathway, striate pathway, become more complex. So we can detect either spots of light, just in this layer 4. You see the little dots in layer 4? Or we can project something as complex as a bar of light using these layers above layer 4 in the orientations as they're positioned by our orientation layers, depending on where we are detecting the object or light in the receptive field. And then we have horizontal connections between neurons that are not in layer 4 that allow us to have stereoscopic vision. So what does that mean? So that means you can have crosstalk between information from the same eye and information from the opposite eye, so that you can visually perceive in 3 dimensions. So the stuff that's coming into one eye, like your right eye, is perceiving the world slightly differently than your left eye is perceiving the world. And you can demonstrate this by covering one eye and pointing to something, or closing one eye and pointing to something, and then opening that eye and closing the other. And you'll notice that the images are slightly different. But what the brain does is it combines both images, and there's crosstalk that allows you to perceive the world stereoscopically in 3 dimensions. So how did they find out about all of this? Some experiments were conducted. And so to this day, if you were to stick an electrode in the brain to see how cells responded to certain characteristics of light, you would find these different response properties throughout the pathway. And so if you look here, in a case where you have an electrode going through all 6 layers, so perpendicular to this peel surface. So the pia is that outer surface of the brain, and in this case, the visual cortex. It will show sensitivity to the same orientation, right? Because remember, those orientation columns have the same orientation all the way down. But if you angled the probe so that it goes through different columns, it will show sensitivity to progressively different orientations. All right, so cells in the retina, thalamus, and layer 4 of the primary visual cortex would respond best to circular spots of light with an on-off and a center surround, so center donut surround antagonist relationship. So you can get excitation when light is shining on the area of maximal intensity, and rebound excitation when the light shines outside, or when you have light turning off outside of that. So cells in other layers of the primary visual cortex are going to have their own designations, own responses accordingly. So you can have simple cells that are going to respond best to bars of light. Okay, and again, this is outside retina, thalamus, layer 4 of the visual cortex. So they're going to respond best to bars of light at specific orientations in on-off regions. You can have complex cells which respond best to bars of light within an entire receptive field. And you can also have hypercomplex cells that are going to respond to specific sizes of bars of light. So is it this size, maybe it's this size within an entire receptive field. All right, so how does this all work? So there is a hierarchy that's thought to arise from the convergence of inputs from progressively more complex cells within the pathway. So for example, you could have these concentric cells. Okay, remember the little dots with on-off, surround, central, distinguishing responses. You can have concentric cells like these little star-shaped stellate cells with the number 1 converging on a simple cell, like this pyramidal cell designated as number 2, and therefore gain a simple receptive field. You can also have simple cells like these pyramidal cells denoted by number 2 in image B, converging on a complex cell like this larger pyramidal cell number 3, and therefore get a complex receptive field. And so on your own time, if you're interested, you can watch this video of the experiment where this guy is looking at a cat and what the cat is seeing on a screen. And basically what he's doing is he's got this image of a dot, and he's moving it out of the cat's line of vision. And then as he moves the paper image, the line of the paper is crossing the cat's visual field. And so the cat is detecting that bar of light, and it's triggering those neurons to fire. And so this is essentially how they discovered that you have these different orientation patterns of neurons that can detect different orientation of dots or bars of light. That is the end of the story. So we continue to process visual information after the visual cortex. This continues into what is known as the extra striate cortex because it is outside of the striate or primary visual cortex. But it is also related to that magnocellular and parvocellular information that we've already discussed. So all of that information is still traveling to these extra striate areas. All right, so this looks a little complicated, but we're going to kind of break it down into components. So where is a stimulus coming from? So this is more so the dorsal pathway or the more superior pathway. And cells in this pathway respond best to moving stimuli. So if they're responding best to moving stimuli, is that the magnocellular or parvocellular?
Is it the magnocellular?
Yep, exactly. Awesome. Think moving magnocellular. All right, so we're going to incorporate our middle temporal lobe, our middle superior temporal lobe, and our posterior parietal. Posterior parietal cortex, and all of these are part of the cortex of the brain. They're just different portions of the cortex of the brain. So that posterior parietal cortex, we can see in image A, designated by the black lines in the white field of space. All right, now what is a stimulus? What a stimulus is, this involves the ventral pathway. So this makes sense because now we're going to bring in more detail, the shape, the color. How do we really identify what something is? We need to pay attention to the detail. So is this magnocellular or parvocellular?
Be parvocellular.
Yep, this would be the parvocellular stuff. All right, so now we're incorporating our inferior temporal cortex and our occipitotemporal cortex. Now, where are those located? If you look at the inferior surface of the brain, here you can see those areas here. You don't really have to distinguish, but you know, the one from the other because they're just really close together, honestly. Now, the specific area within the ventral stream, which we just mentioned, which we just talked about, is related to face recognition. And we have conducted studies through functional MRI or fMRI that use blood oxygen level dependent signals, or BOLD, to look at neural activity indirectly. And so we have figured out that this is where we have our ability to be able to recognize people's faces and see, you know, recognize all the detail of people's faces. So lesions to this area can actually create what's called prosopagnosia. It's really interesting. So watch this video. And this lady cannot recognize her own mother's face. She can't even recognize her own face when she's shown a picture of herself because she's had a lesion to this area. All right, I'm going to skip this because you really don't need to know this. You're not going to be asked to identify where these are in brainstem slices. The whole point of having these slides was just so you could see, have an idea of how these anatomical structures are oriented in different slices of the brain, whether you're in an axial slice like this, so as if you're cutting the brain into superior and inferior halves. So going through, you can see the optic nerve, optic tract, lateral geniculate nucleus in the thalamus, optic radiations on their way to the primary visual cortex. And then you have coronal slices. So if I'm cutting the brain into anterior and posterior slices, we can also see these anatomical structures from that vantage point. All the way back to lateral geniculate nucleus, optic radiations, and more optic radiations until you get to the primary visual cortex. So this image really summarizes what happens when you have lesions along the pathway. So for instance, if I have a lesion that impinges or cuts my optic nerve, regardless of what eye it is, I'm going to have blindness in that eye. All right, but what happens when I have something like a pituitary tumor that impinges on my optic chiasm? Well, because optic chiasm involves those crossed fibers, well, now you're knocking out the hemifields, the the left hemifield in the left eye, the right hemifield in the right eye, and you're getting what's called bitemporal hemianopia. But what if you're just nicking the fibers, the temporal fibers that are on the same size that aren't crossing? Well, now you're only going to lose a hemifield in that affected eye. So you don't have any involvement of the other eye. You just lose a hemifield in that particular eye. All right, but what if you get an optic tract? Okay, so now you have crossed fibers from the opposite eye and uncrossed fibers from the same eye. So now you're going to get loss of the same hemifield in both eyes. So we have left homonymous, because it's the same hemifield in both eyes, hemianopia. So you can kind of go through all of these little diagrams here, looking at where your lesion is localized along the pathway, and see what sort of visual deficits you're going to get. All the way to the visual cortex, where you'll see quadrant deficits at this point, because remember, not only do you have left and right cross fibers of hemifield information, you also have up and down crossing. So right is left, left is right, up is down, and down is up. So I'm going to stop there, and you just won't be responsible for anything beyond this point. But this slide really sort of nicely summarizes the main aspects of your visual deficits anyway. The remaining stuff in this lecture touches on stuff that's going to be covered in other lectures later. So I talked about those non-lateral geniculate nucleus projecting retinal ganglion cells. Some are going to be involved in circadian rhythm. Dr. Syed's going to talk about that, I believe, in Unit 3 when y'all get to regulatory pathways. You also have visual pathways that are associated with involuntary eye movements. So saccades and horizontal vertical gaze, focusing on an area for a prolonged period of time, or involuntary looking at something, tracking something in front of you that's moving. So that certainly involves visual pathways. It's just different visual pathways from the ones we've talked about today. And those are going to be covered in the next unit when y'all get into motor systems, because now you're going to have to involve actual movement of the eye because of those visual pathways and their connections to the nuclei that control the extraocular muscles of the eye. All right, are there any questions?
So to clarify on the columns, so like all of the LGN layers, they project to layer 4?
Yes. And they do it, differentially based on those dominance columns that you see in that diagram.
Okay.
All right, anything that didn't make sense or that was unclear? I know the visual system's kind of complex.
I think I just need to digest everything.
What's that?
I said, I think I just need to digest everything.
Okay. And I told you all that I would give you some practice questions. So hopefully later today, I will have a link to some practice questions that are involving the auditory system. And you can go to that site. Keep in mind that some of these practice questions, you know, were not covered in my actual Zoom lecture. So just note that you won't be responsible for those questions. Just kind of focus more on the ones that we talked about. But those are going to be in a format that will be similar to the questions that you'll see on the exam. And then what I'll also do is later on, I'll try to give you some sample questions for the visual system that you can work with. All right, if there are no questions, I don't know if Dr. Syed needs to say anything or. Well, I guess y'all can just leave.
Thank you.
Well, thank
Okay.
you.
Yeah, no problem.
So, Dr. Meyer, you are done, right?
Right, yeah. I just didn't know if I I didn't want to close something.
No, it's okay.
Okay.